Previously, we looked at the genetics or the mode of inheritance of the G6PD enzyme. In this lesson, we shall be looking at the physiology or the mechanism by which the G6PD enzyme is able to confer protection to the membrane of the red blood cells or the other cells of our body from becoming damaged by oxidants or exposure to free radicals. Now, we want to explain this in a diagrammatic form. And so, this is a metabolic pathway that is known as the pentose phosphate pathway. In fact, this particular pathway is catalyzed by the G6PD enzyme. In other words, the G6PD enzyme is involved in a metabolic pathway known as the pentose phosphate pathway, PPP. The starting material or the substrate for this particular metabolic pathway is the glucose 6-phosphate, which is abbreviated as J6P. J for glucose, then P for phosphate. Now, the first step in this particular reaction is a dehydrogenation reaction. Dehydrogenation simply means the removal of hydrogen. This simply means that this reaction is an oxidation reaction. Recall that oxidation, apart from being defined as the addition of oxygen and the removal of electrons from a species, oxidation can also be defined as the removal of hydrogen atoms. So once there is removal of hydrogen atoms from a molecule, that molecule is said to be undergoing an oxidation reaction. So the glucose 6-phosphate will be undergoing an oxidation reaction, specifically a dehydrogenation reaction. Now, in the nomenclature of enzymes, the enzymes are named with respect to the type of reaction they catalyze and the substrate they are acting on. Since this chemical reaction is a dehydrogenation reaction, all enzymes that catalyzes dehydrogenation reaction are collectively referred to as dehydrogenases, named after the type of reaction. So dehydrogenation reaction is catalyzed by a dehydrogenase. Now, the enzyme is named after the type of reaction and the substrate. The substrate in this context is glucose 6-phosphate. So to get the full name of the enzyme, we add the reaction enzyme name to the substrate. So the reaction enzyme name is dehydrogenase, so named after dehydrogenation. And the substrate name is glucose 6-phosphate. Now, dehydrogenase is abbreviated as D. And so once we add dehydrogenase to glucose 6-phosphate, then the name of the enzyme catalyzing this reaction becomes glucose 6-phosphate dehydrogenase, abbreviated as G6PD. The D here symbolizes or stands for dehydrogenase. So the G6PD, that is the glucose 6-phosphate dehydrogenase, having looked at how the enzyme name has been established, we know that the enzyme is catalyzing a dehydrogenation reaction, which means that there is going to be the removal of hydrogen atoms. And that implies that there must be a species to accept these hydrogen atoms. And that will bring us to the species called NADP+. That is nicotinamide adenine dinucleotide phosphate. Once you see the plus, it symbolizes oxidized. So this is the oxidized nicotinamide adenine dinucleotide phosphate. So the G6PD as a dehydrogenase enzyme catalyzes the removal of hydrogen atoms from glucose phosphate, and these hydrogen atoms is added to NADP+. So the NADP+ accepts the hydrogen to become NADPH. And so the NADPH, in other words, is reduced to NADPH plus H+. Now, this means that this direction is a reduction reaction, because we define reduction as the addition of hydrogen to a species, whilst oxidation is the removal of hydrogen from a species. Once the hydrogens are being removed from glucose phosphate, removal of hydrogen from glucose phosphate implies that glucose phosphate is undergoing an oxidation reaction. And the hydrogen atoms that has been removed being added to the NADP+ implies that NADP+ accepting the hydrogen implies that it is undergoing a reduction reaction. So this is a typical redox reaction because both oxidation and reduction are occurring simultaneously. This is where in the qualitative screening of the G6PD enzyme, the reagent called methylene blue comes in. The methylene blue actually activates the pentose phosphate pathway. Specifically, it activates the enzyme G6PD. And so once it activates the G6PD enzyme, the G6PD will be involved in the pentose phosphate pathway, and simultaneously oxidation and reduction will occur. That is a redox reaction. For this reason, the methylene blue is named as a redox dye because it stimulates an oxidation and reduction reaction by activating the G6PD enzyme. So the methylene blue is called a redox dye because of its activation of the G6PD enzyme and the pentose phosphate pathway. Now that we have formed the NADPH, which is the reduced form of NAD+. Now this reduced NADPH now reacts with GSSJ, which is oxidized glutathione. So NADPH reacts with oxidized glutathione, converting the oxidized glutathione to reduced glutathione. So because conversion of oxidized glutathione to reduced glutathione is a reduction reaction, notice that reduced glutathione has hydrogen, which is being obtained from NADPH, whilst oxidized glutathione does not have hydrogen. So this NADPH serves as a donor of hydrogen to the oxidized glutathione, converting the oxidized glutathione to reduced glutathione. So this is typically a reduction reaction. From oxidized glutathione to reduced glutathione is a reduction reaction. So all reduction reaction in the nomenclature of enzyme, all reduction reactions are catalyzed by the enzyme called reductase. So the reductase is the enzyme that catalyzes a reduction reaction. So there is going to be the conversion of the oxidized glutathione to the reduced glutathione. So that reaction is catalyzed by a reductase enzyme because the reductase is actually involved in a reduction reaction. So once the oxidized glutathione is converted to the reduced glutathione, simultaneously the reduced NADPH also becomes converted to NAD+, because the reduced NADPH will donate. There is removal of hydrogen atom from NADPH, which is being added to oxidized glutathione. Whilst the oxidized glutathione is being converted to the reduced form, the NADPH, once it loses the hydrogen, that is removal of hydrogen, then tends to be an oxidation reaction, regenerating the original NADP+. Because this reaction that converted the oxidized glutathione to reduced glutathione is a reduction reaction, and the name of the enzyme is reductase, and the molecule that it is reducing is called glutathione. The name of the enzyme named after the reaction type, and then the substrate will be therefore glutathione reductase. So the glutathione reductase catalyzes the conversion of oxidized glutathione to reduced glutathione. So now that we form the reduced glutathione, so this typically again is an oxidation, reaction and a reduction reaction, another redox reaction. So now that we form the reduced glutathione, this reduced glutathione, which happens to be the most potent or powerful antioxidant in the cells of the living system, will now act on reactive oxygen species such as superoxide free radicals, and these are generated during mitochondrial respiration, drug metabolism. Exposure to radiations generates free radicals, and these free radicals, commonly superoxide free radicals, they can become converted to hydrogen peroxide, all of which are reactive oxygen species that needs to be neutralized, else they will cause damage to the cells of our body through the process of oxidation. So these reactive oxygen species are what we classified as oxidants. So the oxidants will cause oxidative damage to the lipids, the proteins, and the DNA of our cells. To protect these cells from becoming damaged through oxidative processes, the reduced glutathione being formed under the influence of the enzyme GSPD now acts on these oxidants or free radicals, converting these reactive oxygen species, specifically convert hydrogen peroxide to water. And this happens under the influence of the enzyme known as glutathione peroxidase. As the name implies, a peroxidase is an enzyme that acts on peroxides, and the molecule, the reactive oxygen species, is hydrogen peroxide. So the glutathione peroxidase here converts the hydrogen peroxide to water. And once there is the conversion of hydrogen peroxide to water, the reduced glutathione loses its reduced property and becomes converted to oxidized glutathione, awaiting for the production of reduced NADPH from the activity of the GSPD enzyme, that is removing hydrogen atoms from glucosase phosphate and adding it to NAD+ to form NADPH plus H+, which will in turn combine with the oxidized glutathione to form the reduced glutathione. Now, in the qualitative screening test for the GSPD enzyme, the other reagent, apart from the methylene blue reagent that is being used, which activates the pentophosphate pathway, and of course, it is activating a redox reaction and is so named as the redox dye. The other reagent is called the sodium nitrite. And the sodium nitrite comes in because we are trying to take an advantage of what actually happens in the cells of the living system, which is when the cells are exposed to oxidants, the cells will undergo the components, or the proteins and lipids will undergo oxidation. So what happens is that we will intentionally expose our red blood cells to oxidant. So in the laboratory, we have the sodium nitrite acting as an oxidant, which will cause oxidative stress by converting the hemoglobin molecules that are contained in the red blood cells to methemoglobin. So the methemoglobin becomes the oxidized version of hemoglobin. So these two most important reagents, the methylene blue acts here, that is the redox reaction, whilst the sodium nitrite reagent is serving as the oxidant. So that is how the principle for using the methylene blue reagent and the sodium nitrite reagent.